Today's lesson is a continuation of enzymes, and today we're going to talk about different factors affecting enzyme function. There's a wide variety of them. We have enzyme concentration, substrate concentration, temperature, pH, salinity, and then we're going to talk about activators and inhibitors. Enzyme concentration is going to affect the function of an enzyme because as you have more enzyme, then you're going to have a higher reaction rate at first. The more enzymes there are, the more frequently they will collide with their substrates and give you more reactions. After a while, though, the substrate becomes a limiting factor. As you have more enzymes, that doesn't mean you have enough substrate to work with every single one of those enzymes, so your graph is going to level off like this. The same thing happens with substrate concentration. The more substrate you have, the more frequently it will collide with the enzyme, but at some point, all of the enzymes are going to have their active sites engaged. That means the enzyme has become saturated, and there's still more substrate floating around. The little yellow circles are the substrates, but the enzymes are all busy, they're all full, so you're going to have a leveling off, and this maximum rate of reaction has been achieved. Temperature is another factor that influences enzyme function, and generally each enzyme has what's considered to be its optimum temperature, which is the greatest number of collisions that happen at that particular temperature. So it's the peak of the graph here, and heat can increase the energy level of the molecules. It's going to disrupt the bonds that form. Remember, enzymes are proteins, so when we heat things and we disrupt the bonds in the enzymes, you are going to have a reduced ability of that enzyme to bind with its substrate, and at some point, that enzyme will denature. It will lose its three-dimensional shape or its tertiary structure, and it will be unable to work anymore with that particular substrate. So the shape, remember, the shape of the enzyme is very important to its ability to be able to do its job, and heat will disrupt the bonds that form there, and it will change the shape of your protein, making it unable to work. Some enzymes can renature after they have lost their shape temporarily. Other enzymes, once they are denatured forever, and they will never go back to how they were before. Cold, on the other hand, just makes the molecules move slower, so you have less collisions. So you're going to have a lower reaction rate. The enzymes are not going to work as well at cold temperatures, but it's not due to them being denatured. Heat is the only thing that denatures enzymes. Cold just makes everything go slower. So here's a couple examples. Different enzymes function at different optimum temperatures. So you can see here, we have a human enzyme that would work best at 37 degrees Celsius. That is the internal body temperature of a human. And then here we have, like at Yellowstone, when the geysers and hot springs, the bacteria that live inside those hot springs and geysers have a much higher optimum temperature where they work. And you can see that for them, a cold temperature where they have a slow amount of reaction here is actually much hotter even than our internal body temperature. So you can see the point where the enzyme is denatured and it stops working. And you can see where it's a slow rise at the beginning because it needs to warm up. Those cold temperatures make it go slower. pH changes can also disrupt the bonds within the different structures of the protein. So we can, changes in pH either add or remove hydrogen ions, and those are going to disrupt the bonds that occur. It's going to affect the secondary and the tertiary structures of those proteins and cause them to denature, right? We have charged amino acids that when you add another charged molecule to it, you're going to change the configuration. You're going to change the bonds that form. And just like with temperature, we have optimal pHs For most human enzymes, it's somewhere around neutral, which is 7. So the red one here would be human. So that would be 6 to 8. But then in other locations in our body, like our stomach, for example, the enzyme pepsin works best. That's the green one here, at a pH between 2 and 3, because that's acidic. That's like our stomach acid. That's where it works the best. And then in our small intestines, it's actually a basic environment to counteract the acidity from our stomach. So trypsin is an enzyme that's in our small intestines, and that one has an optimum pH around 8 or so. So you can see that each one has a very narrow range of where it works best. Salinity, which is salt, the salt concentration, can also disrupt the bonding structure and denature a protein. So again, adding or removing positive or negative ions is going to disrupt that structure of those proteins. It's going to disrupt the attractions between different charged R groups on those amino acids, and it will denature proteins. Enzymes are very intolerant of extreme salt, and this is just kind of a side tidbit here. The Dead Sea is called dead for a reason. There's so much salt in the Dead Sea that very few enzymes can actually work there, and enzymes facilitate all the chemical reactions for living things, so there are not very many living things that are able to survive in the Dead Sea because of the salt. We have activators. Activators are compounds which help enzymes out. There are 2 different kinds. There are cofactors, which are non-protein based inorganic compounds. So you have things like magnesium, potassium, calcium, zinc, iron. Those are all considered to be cofactors. And if you look over here, these 2 pictures are both examples of cofactors. In the center here, you can see that magnesium atom, right in the center of the chlorophyll molecule for photosynthesis. That is a central component of it. And the same thing with the hemoglobin enzyme. Those proteins need to have iron molecules in them. It gives them a higher affinity for the oxygen that they carry. So those are cofactors. Coenzymes are also non-protein based molecules, but they are organic molecules. They bind temporarily or permanently to the enzyme near the active site and just make the enzyme work better. A lot of our vitamins work this way. Vitamin B3, B2, coenzyme A, those are all considered to be coenzymes where they help make the enzyme work better. We have other inhibitors. These regulate the enzyme activity. They actually reduce the ability of the enzyme to work. And we're going to talk about all of these here in the next couple of slides. A competitive inhibitor is an inhibitor that competes for the active site. It's something that will actually go in here and it will block the active site of the enzyme so that the substrate is unable to bind to the active site there. There are a couple different medicines that are examples of that. Penicillin is an example. It actually blocks the enzyme that the bacteria use to build their cell walls so that they cannot build walls properly. So they can't replicate if they can't make their full surrounding. There's a couple examples there. Sometimes they can permanently bind to the active site, meaning that it is irreversible inhibition, and other times it's temporary. A non-competitor inhibitor binds to a different place on the enzyme. It's called an allosteric site. So allosteric site means that it's not the active site. It's someplace else on the enzyme. But what happens is it changes the shape of the enzyme and it changes the shape of the active site so that the substrate can no longer fit into the active site of that particular enzyme. There are some anti-cancer drugs that will do this. They will inhibit the enzymes involved in DNA synthesis so that you can't make more cells. Cyanide poisoning is actually an irreversible inhibitor of a cytochrome C enzyme that happens in cellular respiration, where you can't make ATP anymore, so your body can't make any more energy. Again, if it's a permanent binding, then it's irreversible inhibition, but most of the time it's temporary. This allosteric inhibitor will leave, the enzyme will go back to its normal shape, and then the substrate can work again. Feedback inhibition. These are just pathways that happen in our bodies. They are for the regulation and coordination of different processes. And you can see in this one, you have the initial substrate, and then the substrate binds to this one particular enzyme, and then that creates another molecule, which then binds to another enzyme, and it keeps going on and on and on until you get to this end product. What often happens with these feedback loops, they're called, is that the end product is actually an inhibitor of an enzyme earlier on in the process, so it stops the process. If you have enough of this isoleucine right here, that means that your body does not need to make more of it, so it's actually going to come back up here and it's going to inhibit the enzyme from working. So that means that you cannot go through this process again to make more isoleucine. Once your body needs that last isoleucine, that's going to be used, and then it's going to go back to its normal functioning to make more of this product. Okay, so these are feedback loops. We have a kind of a domino effect here, but the end product will inhibit an enzyme earlier in the process, because if you don't need to make it, your body isn't going to waste the effort in making more of it until it needs it. And that is it for enzymes.